Bernard said, I changed a bit the title because while I was preparing this talk for today, I thought it would be more interesting for you to experience two different studies which demonstrate how native mass spectrometry can be used in order to study the structure function relationship of large protein complexes. So I decided to change the title and divide my talk to two. And in the first part of my talk, I will demonstrate how we use the native mass spectrometry approach in order to develop an approach to define the allosteric mechanism and function of large protein complexes, especially ligand-driven protein complexes. And in the second part of my talk, I would like to demonstrate how we use this method to discover a new subunit for an old complex, and I'll demonstrate how we discovered the existence of the ninth subunit for the signalosome complex. So let's start with the first part of my talk, which focuses on a method for defining the allosteric mechanism of function of ligand-driven protein complexes. And this was a joint study in collaboration with Professor Amnon Horvitz, Ranit Gruber, a graduate student in his lab, and Andrei Dachenko, which was a visiting student in my lab for 6 months, and he's currently in Albert Hecht's group. So as you probably know, one of the most important ways to regulate the function of a protein complex is through ligand binding, because ligand binding can induce conformational changes that will either activate or deactivate a protein complex. And many theories were developed in the late 60s in the previous century to try to explain how allosteric mechanism is taking place. And the common thing between the different models was that they assumed that subunits within a protein complex can exist in either of the two forms. A subunit can be either found in the T form, which is a tense state, which has low activity and low affinity for the ligand, or it can be found in the R state, which is a relaxed state that has high activity and high affinity to the ligand. And the difference between the different models was how this structural transition between the T to the R state occur and what are the intermediate states. And like I've said, many theories were developed, but the most common and classical models to describe allosteric mechanism of function are the MWC model, which is also called the concerted model, or the KNF model, which is also called the sequential model. And I won't go into the deep details of these two models. I just want to very briefly demonstrate the difference between them. So according to the concerted model, the entire protein complex undergo a concerted conformational shift from the T to the R state. So for example, here the T state is shown in squares, the R state in circles, and when the ligand bound, the color is changed from orange to green. So we start usually when all the subunits are found in the T state, they have low affinity for the ligand. However, once the ligand is starting to bind to the protein complex, due to the high affinity of the ligand to the R state, the entire protein complex undergo a concerted shift in structure to the R state. According to the KNF model, the structural transition can happen in a sequential step. So once a ligand is bound to the complex, a subunit is converted between the T to the R state, and we have all the intermediate compositions in the middle. So although these classical models for describing allosteric regulation were suggested more than 50 years ago, it was actually impossible to distinguish between them experimentally and to accept or reject one of the models. And the main reason for that is that most techniques actually look at the ensemble of coexisting ligand states, and they could not differentiate between them. And we were thinking that maybe mass spectrometry can fill this gap. Maybe we can take advantage of the fact that within a single spectrum, you can actually identify all the coexisting states, and therefore you can determine their relative abundance, and therefore this could be a good method to distinguish between the two allosteric models. And as a model system, we have chosen to work with GOEL because GOEL has become a paradigm for studying allosteric machines. This is a bacterial chaperone. It is composed of 2 rings and 14 identical subunits. There are 7 subunits in each ring, and each one of the GOEL subunits binds ATP. And if you look at the mass of GOEL, which is 801 kilodalton, and you look at the mass of ATP, which is 500 dalton, you can already appreciate the challenge in this study. How can we distinguish between the small shifts in mass that are associated with ATP binding? And if you think about it, it's like counting the number of birds that sit in an elephant by measuring its weight, or something more practical for parents, deciding if your child has lice or not by putting him on the balance. So really the major challenge was here to be able to distinguish between the small shifts in mass. So here is an example of a native mass spectrometry spectrum of GOEL, and if I'll just enlarge this region, you can see how we obtain a distribution of charge states and how these charge states are separated from one another, but we still don't have the resolution that we need in order to be able to count the number of ATP-bound molecules. And therefore, the initial thing that we did, this is something that Andre did, was to screen for a sample solution that we can use that will allow us to get highly resolved peaks. We specifically wanted to use a buffer that will, first of all, reduce the number of charges, because then the peaks will be more separated from one another, they will be more resolved. And we also wanted to use a buffer that will not be bound to the protein complex, that it will give us very fine structured peaks. And also we needed a buffer that will not allow for ATP hydrolysis, because you know when GOEL binds ATP, it undergoes hydrolysis to ADP. And we wanted to freeze only the ATP bound state. So for example, ammonium acetate, which is shown here, this is a spectra that was acquired in the presence of ammonium acetate, was not a good buffer because ATP undergo a hydrolysis to ADP using this buffer. And another complication is that we had our samples to include also 1 millimolar magnesium acetate in order to preserve the cooperative nature of ATP binding. So we're adding our salt to our solution, which of course can also increase the peak widths. So we started by adding imidazole. You can see that imidazole actually induces a charge reduction, and the peaks were more separated from one another, which was good, but this was still not good enough. We still didn't have the resolution that we needed. We then used ethylene diammonium diacetate, which gave further charge reduction. The peaks were really nicely separated, and they had a very fine structure. And we also tried triethyl ammonium acetate, which the peaks were, it gave a really dramatic reduction in the charge state, but the peaks were quite broad because this buffer tends to stick to your protein complex. So we started to stick with ethylene diammonium diacetate. As far as we know, this buffer was not used before for native mass spectrometry. And as you will see, I really recommend it. It gives really nice, fine, structured peaks. So we did a series of titration experiments in which we gradually increased the concentration of ATP from 0 to 100 micromolars. In total, we had 21 different concentrations. And this is an example of a spectrum that was acquired in the presence of 5 micromolars ATP. And you can see the structure that the peaks are starting to have. They are splitted to different numbers, and this is due to the binding of ATP. And if I'll just enlarge one of the peaks, for example, the 58 charge state, you can see that we actually have 5 peaks representing the apo form of GOEL, but also GOEL bound to 1, 2, 3, and 4 molecules of ATP. And then we could use a deconvolution approach and calculate the area below each peak, which actually represents the relative abundance of all the species. we could sum up the deconvoluted peaks, and you can see that the sum up spectrum, which is shown here in black, fits very nicely to the measured data. And this is an example of only one chart state, but actually we can use all the different chart states. The different chart states are like a replica of the same experiment. So this is exactly what we did. Here you can see all the different chart states for the previous spectrum I've shown you. Here on the x-axis are the different bound ATP molecules from 0 to 4 and their relative abundance, and on the top is the standard deviation between the different chart states. And you can see that the standard deviation are really low. So what we actually used is the average relative abundance that we obtained. And then we could really follow how ATP molecules are gradually bound to GOEL. And now I will show you multiple superpositions of different spectra that were acquired in increasing concentration of ATP. So we start with the APO form that has no ATP molecules associated with it, and then we can gradually increase the numbers of ATP bound states. And we could really count in each concentration how many ATP molecules are bound. And you can see that this big jump that we have here is due to the cooperative nature of ATP bounding, which is known to exist for GOEL. So it really showed that we can count the number of birds that sit in an elephant by measuring its weight. But you know, science is never straightforward. And at this step, we also discovered that we have non-specific binding. And this is something that often occurs in the electrospray method, that due to the desolvation process, you actually increase the concentration of the ligand, and therefore you have non-specific binding. So we had to clear our data from the artificial increase in intensity due to non-specific binding. We search in the literature for different known approaches that allow you to remove non-specific binding contribution. John Klassen, for example, has a method in which you add a reference protein that is known not to bind your ligand, and then you count the number of non-specific bound ligands to this reference protein. However, unfortunately, this method was not relevant for GOEL because GOEL is a very friendly protein, and no matter which reference protein we added, it was always associated with the GOEL. In other methods, you had to assume that you have a non-cooperative system, which was not relevant to our case. So at the end, we actually developed our own method, which is it's a generic method to remove the contribution of non-specific binding, but you do have to know the number of specific bound sites. So just for simplicity, I'll explain the essence of this method, and for simplicity, let's assume that we have a dimer. So if we mix our dimer with a ligand and we measure the mass, we can either measure the mass of the APOR dimer, we can measure the mass of a dimer bound to one ligand, but this could be due to specific binding or to non-specific binding. We can measure the mass of the dimer bound to two ligands, either both of them are bound specifically, one bound specifically and one not, or both of them are bound non-specifically. So based on the mass spectrometry data, we can extract the non-specific binding constant value. We assume that there is a single non-specific binding constant, and then we use this value in order to subtract the artificial increase in intensity due to the non-specific binding, and we add this intensity to the relevant group. For example, we remove this intensity and add it to one ligand bound specifically. And this allows us to correct our data. And this is exactly what we did with our GOEL system, and here is a summary of all the results. So on the x-axis, you have the different bound states of GOEL, starting from the APO form to the fully occupied GOEL. Here on the y-axis, we have the 21 different concentrations, and here on the z-axis, we have their relative abundance. So we start with the APO form of GOEL, which goes down, and we finish with the fully occupied GOEL, and in the middle we have all the intermediate states. And now that we have this information, we can go back to our original question and use this data in order to define the allosteric mechanism and function of GOEL. So one of the things that we could do was to determine the Hill coefficient as a function of the ligand concentration. The Hill coefficient determines the extent of cooperativity, and usually a single value is used, and this is because the standard methods allow you to extract only a single value. However, it was known theoretically that the Hill value should change as a function of the ligand concentration, and here we could follow it. We see that at low ligand concentration, we start with a value of 1, which shows that there's no cooperativity. The Hill coefficient then goes up to a value of 2.3, and then it goes back to 1. And this increase and decrease in the value of the Hill coefficient was suggested, like I've said, and it was shown experimentally only for hemoglobin, because hemoglobin binds oxygen under very wide distribution of concentrations, 4 orders of magnitude. And this was the only system that it was possible to measure it. But now, by using mass spectrometry, this can be actually a method that can be done for any protein complex. Another thing that we were able to do was to define the 14 different binding constants of GOEL. So here you can see the different binding step, and here the y-axis is the binding constant value. And you can see that there's positive cooperativity because we start with low values and then they go up very strongly. And to cut a long story short, what we actually did is that we assumed that GOEL follows the MWC model, meaning that it starts in the starting position, both wings are found in the T state. Due to ligand binding, one wing and then the entire wing is converted to the R state, and due to the additional ligand binding, the second wing is also converted. And if this is indeed true, we calculate a model that represents this, and you can see that the black line is the model. And there's very good agreement between the model and the values that we could extract from the mass spectrometry method, suggesting that indeed GOEL follows the MWC model. And if it follows the MWC model, we actually have all those states. So we either have the TT state, in which both wings are in the T state, we have the TR state, in which one wing is already converted, and we have the RR state, in which the second wing is also converted, and for each state we have the different numbers of ligand-bound states. And now that we have the values from the model, we could actually calculate the relative abundance of these different states. So if a population is found between 20 to 100%, it is colored dark green. If it is between 1 to 20%, it is light green. And if it exists less than 1%, it has no color. So now we could also see that it is enough for 3 molecules of ATP to bind to GOEL for the entire wing to go through the structural transition. And for the second wing, we need at least 5 molecules of ATP to bind in order for it to go this structural transition. So not only that we could determine the model, but also see exactly how the ligand binding step occurs. And while this system was applied for GOEL and ATP, it is a general approach, and it can be used to any ligand-driven molecular machine, no matter if it binds a drug, a cofactor, a drug, and so on. So this was one way we could use the native mass spectrometry approach for structure-function relationship studies. And now I would like to show you another example that focuses on how we discovered a new subunit for an old complex, and this was the graduate studies of Shelly Rosen, which is now a postdoc in Boston, and assisted her in the biological aspects are Maria and Gili. And the protein complex I'm referring to is the COP9 signalosome complex. This complex regulates the ubiquitination process. As you probably all know, proteins that are damaged or misfolded, or regulatory proteins that need to be eliminated fast from the cell, are tagged with the polyubiquitin tag and degraded by the 26S proteasome complex. And the signalosome complex is a complex that regulates the ubiquitination process. Specifically, it regulates the Kuhlin wing ligase family, the CRLs, and this family contains more than 200 E3 ligases. And these ligases are important for targeting 20% of the proteins that are degraded by the proteasome complex. And among them are many cell cycle regulators and many proteins involved in tumorigenesis, and therefore the signalosome complex is considered to be a very important target for cancer therapy. And it was accepted for many years that this protein complex has 8 different subunits called CSN1 to CSN8 according to their decreased molecular weight. And what I would like to show you actually that this complex has a 9th component, which we call CSNAP for CSN acidic protein. Unfortunately for us, the name CSN9 was already taken, so we couldn't use it. And this is a very small protein. It is 6.2 kilodalton, and therefore this is probably why it was not discovered before, even though this protein complex was studied for more than 20 years. It probably escaped the SDS gels. In addition, it has very low, it has only 2 lysines, so it is hardly stained by Comasi gels. And also in proteomic analysis, it gives very long peptides that are not always identified by standard proteomic methods. So how did we came across CSNAP? So when we started this project, our aim was actually to examine the multiple form of variants of the signalosome and subunits. And in order to do that, we applied a method that we recently established in the lab, in which you take your protein complex of interest, you denature it under denaturing condition to separate the subunit building blocks from one another. We then use a monolithic column that we synthesize in the lab to separate the subunits from one another, and then diluted flow is split to 2. One fraction goes directly to the mass spectrometer, and this allows us to measure the intact mass of the subunits. And then we can actually determine how many coexisting states of the same subunit are present. The second fraction is fractionated into a 96 well plate where it is subjected to proteomic analysis, and this allows us to determine the identity of the protein subunits and also their post-translational modifications. And then we can actually correlate the information that we get from the top down and from the bottom up approaches. And this is an example how the system looks like. So we have the nano-LC system. The monolithic column is placed here. It goes to the nanomate robot, which takes 1/3 of the flow directly to the Q-TOF instrument, and 2/3 of the flow is fractionated into a 96 well plate. This is actually an example how different parts from different mass spectrometry companies can live in harmony together. And here is a representative chromatogram of the signalosome complex that we have isolated from human red blood cells. And you can see that we could separate all the subunits from one another, and we discovered that almost all the signalosome subunits have more than one form. So actually, when we are talking about protein complexes, we don't have a uniform protein complex, but we actually have an ensemble of this protein complex due to the assembly of the different forms of the subunits. And I have to say that this is something that we see also for all the other protein complexes that we have studied. And I'll just demonstrate this for 2 subunits. So for example, let's start with the previous one. So for example, CSN7 is known to be encoded by 2 different genes, CSN7A and CSN7B. And we could see from the proteomic analysis that both CSN7A or CSN7B can be incorporated within the complex. And by the intact mass measurements, we could actually determine the relative abundance of these 2 states. We could see actually that CSNB is a dominant subunit, and it is interesting actually to follow this and determine whether different tissues have different relative abundance of the 2 subunits, and whether different stimuli induces one subunit or the other, and to start to understand why we can have a signalosome either with CSN7B or CSN7A. What's the functional difference between the 2? Another example is CSN8, so we could identify from the proteomic analysis that the subunit that elutes after 17 minutes is CSN8. And again, we could see that there are 2 different forms of CSN8. The higher molecular weight form, which is shown here by triangles, actually fit the intact mass of CSN8, missing the first methionine. But the question was, what is the lower molecular weight form? And if you look at the sequence of the CSN8, you can see that it actually has a nearby methionine, methionine 6, and the lower molecular weight form is a product of a second translation initiation site. So similarly, we could identify that also CSN2 has an alternative translation initiation site, and we could identify that different subunits have different post-translational modifications. But what really attracted our attention was the fact that in all our analysis, there was an additional protein that continuously appeared in our LC chromatograms and mass spec analysis. And this protein eluted after 8 minutes. According to the proteomic analysis, this was a myeloma overexpressed protein number 2, MYEOV2, which I'll now on refer to as CSNAP. And according to the intact mass analysis, its mass was 6.2 kilodalton. However, just the fact that CSNAP co-eluted with a signalosome subunit does not necessarily indicate that it is a specific protein interacting partner. And in order to further validate this, we have isolated the signalosome complex from a different source. And in the case of the human red blood cells, we used a multiple-step biochemical purification method. In the case of the HEC293 cells, we used cells that stably expressed a flag tag CSN2 subunit, and we used a single affinity purification step. So the fact that we were using two different systems and the fact that we were using two different isolation methods actually suggests that we will not have identical contaminants. So the fact that CSNAP appeared again suggests that it specifically interacts with the signalosome subunits. But in order to further validate this, we applied the native mass spectrometry approach, and here you can see the mass spectrum of the signalosome isolated from the HEC293 cells. And you can see that the peaks are quite broad, and this is due to the biological heterogeneity of the signalosome complex that I just mentioned in the previous slides due to the coexisting variants of identical subunits. So just by measuring the mass, we could not determine unambiguously that CSNAP is indeed a specific binding partner of the signalosome. And in order to overcome this, what we did is we applied the MS-MS approach, in which we selected a specific ion, and we then applied the CID method. So when you do CID, a native mass spectrometry, you activate your protein complex due to the collisions with the background gas. We use argon, and due also to the accelerating energies, that the protein complex is excited until a certain threshold is reached, and one subunit is then dissociated from the complex. And then you are left with an individual subunit that are dissociated and with a stripped complex. So in the next slide, I would like to show you only the individual subunits that are stripped off from the signalosome complex. So here are the peaks that we have isolated, and here is a low M over Z region, which we could see the individual subunits that are dissociated. We could identify CSN6, CSN7, CSN8, and we were very excited that we could also see peaks corresponding in mass to CSNAP, suggesting that CSNAP specifically binds the signalosome subunit. At this stage, we also turned to biochemical and cellular methods in order to validate that CSNAP is indeed an integral bona fide binding integral subunit of the signalosome, and I'll just show you a few results from this. One of the first things that we did is that we generated an antibody against CSNAP using the full sequence of this small protein. But here is a place to mention that if you go into the data bank to Uniport, you can actually see that CSNAP has two different isoforms. It has a small isoform which has 57 amino acids, and it is colored here in red, and this corresponds to CSNAP. But it also has a longer isoform which has a molecular weight of 27 kilodalton, and the entire sequence of CSNAP is embedded within this longer isoform. However, using mass spectrometry, it is easy to distinguish between the two, because if you do top-down approaches, they have very distinct mass, and if you do bottom-up approaches, due to this insertion in the middle and to the unique glutamine the CSNAP has, again, it is possible to distinguish between them. So far, all our mass spectrometry analysis indicated that only CSNAP is part of the complex and not the longer isoform, which I call MY2L or MYEOV2L. So one of the first biochemical assays that we did is that we took cell extracts and we separated them on a size exclusion column, and then we looked at the different fractions, and we could see that the signalosome subunit and CSNAP elute exactly at the same fraction, whether the longer isoform elute at lower molecular weight fractions, suggesting again that it is not part of the complex. In addition, we did reciprocal co-immunoprecipitation analysis. Here, for example, we transiently expressed a flag CSNAP, and then we did a pull down, and you can see that together with CSNAP, we also have signalosome subunit pull down. And similarly, in the reciprocal co-immunoprecipitation analysis, when we stably express flag CSN2, and we do a flag pull down, only CSNAP, but not the longer isoforms pull down, again suggesting that CSNAP is part of the complex. And this experiment was repeated also in HeLa cells and also in mouse A31N cells. In all these cases, we got similar results. We then wanted to quantify, after we were convinced that CSNAP is indeed an integral subunit, we wanted to quantify what's the stoichiometry of interaction between CSNAP and the other subunits. And we therefore used the aqua approach. And this was a collaboration with Yishai Levin from the Weizmann Institute Proteomics facility. So what we did is that we synthesized isotopically labeled CSN subunit peptides, and we then quantified this peptide using amino acid analysis, and we spiked those quantified peptides into an isolated signalosome complex. We performed a triptych digestion, and we used the SRM approach. This is actually the aqua method, and then we can quantify each one of the peptides. And you can see that CSNAP actually appears in a 1-to-1 ratio in comparison to the other signalosome subunit, in this case CSN2 and CSN4, suggesting that it is found in an equal molar concentration to the other subunits. And we did a lot of additional biochemical assays. I don't want to bore you with this because I know this is a mass spectrometry course, but we could specifically show by using cross-linking approaches, this is for heading, we could show that it binds to CSN3, CSN5, and CSN6. We could also determine that the C-terminal region is important for incorporation of CSNAP into the complex, and we could also exactly define that the aromatic residues in this region are critical for such an interaction. But we wanted to demonstrate within cells that CSNAP is indeed an intrinsic subunit. And in this case, we did this by taking advantage of the fact that we previously shown that if you induce DNA damage by UV radiation to cells, the signalosome is transiently relocalized to the nucleus, to the chromatin bound and to the nuclear soluble fraction. So if CSNAP is indeed an integral subunit of this complex, we will expect it to follow a similar relocalization. And this is indeed what we see. We see that there's increased amount in the levels of CSNAP following DNA damage in the nucleosoluble and chromatin-bound fraction, and there's no change in the levels of the longer isoform. And the last piece of evidence I would like to show you that we've validated in cells that CSNAP is an integral subunit is that we use the FRAP method, the fluorescence recovery after photobleaching assay, in which you tag your protein of interest with a fluorescent tag. You then bleach a specific region within the cell. We did it both for the nucleus and the cytoplasm, but I will also only show you the results from the nucleus right now. And then you follow how fast there is recovery in fluorescence in the area that you bleached. And this allows you to generate a recovery curves. And you expect that subunits within an identical protein complex will actually have similar recovery curves. And we used different constructs. We used the free cerulean, which is the free fluorescent protein, to represent the dynamics of a fully mobile monomeric protein. We use full-length C-SNAP, which the cerulean is fused to the C-terminal. We use C-SNAP, which is missing the C-terminal, and we use C-SNAP, which is missing the N-terminal. We could previously demonstrate that all the signalosome subunits have a similar mobility in FRAP assays. So here I'm just showing you CSN3 as a representative of a signalosome subunit. And as an additional control, we use DDB2 because DDB2 is a nuclear protein that is known to be associated with a signalosome complex. However, it is not an integral subunit of this complex. So we started by looking at the control of the fully mobile monomeric protein. And you can see that if we remove the C-terminal region of C-SNAP that we already showed by biochemical assays that is necessary for incorporation into the complex, it behaves exactly like a free cerulean. So without the C-terminal region, C-SNAP is not part of the signalosome complex. However, full-length C-SNAP and C-SNAP missing the N-terminal and CSN3 have a similar mobility, which is different from DDB2. Again, showing also within cells that C-SNAP is an integral subunit. And to examine whether this subunit has a biological relevance, after many efforts that we tried to knock down this subunit using siRNA, shRNA, morpholino, all these methods were not successful, we managed to knock it out using the CRISPR system. And we could see that cells that are lacking C-SNAP, these are in dark gray, these cells have lower proliferation rate. In addition, cells that are lacking C-SNAP are much, much larger. This can also be quantified, and you can see that they are more flattened and larger. So this actually showed that C-SNAP also has a biological importance because when you remove it, cells have a distinct phenotype. And the last piece of evidence I would like to connect to another protein complex. The signalosome has a one-to-one corresponding with subunits of the 19S-lid proteasome, suggesting that the two protein complexes actually originated from a common evolutionary complex. And each one of the signalosome subunits can be referred to a 19S-lid subunit. And here you can see the sequence of identity between the human protein subunits. And the only subunit that was missing a counterpart is DSS1. And we suggest that C-SNAP actually fills this gap. Not only that both subunits have an 18% sequence identity, but they are both intrinsically unstructured small proteins. So with this, I would like to end my talk, and I hope I can convince you that the native mass spectrometry approach can be a powerful method for structure function analysis of protein complexes. I've shown you that we use this method to distinguish between allosteric mechanisms, and also this method helps us to discover a new subunit for an old complex. And with this, I would like to end my talk. I would like to thank, all my group members that are now saying hello to you. I would like to thank my funding, and thank you for your attention.